Hello, everybody. My name is Bryce McCourt. I am an online student at Grand Canyon University. I am majoring in chemistry education, and this is my presentation of the cell cycle regulation in the cell for the Concepts of Biochemistry class. So whenever you are presenting in front of students, they would need to know a good introduction, and then they would need to know their learning goals, their learning objectives. What is it that they need to take away from the lesson that is valuable? What is being expected of them to learn so they can understand certain concepts? And so the very first learning goal is that students will identify important key stages regarding the eukaryotic cell division and regulation. Second, the students will be able to understand the importance of specific regulators as checkpoints in the regulation of the cell cycle. And finally, students will analyze the important factors of cell cycle regulation, such as the following: cell size control, DNA damage responses, DNA replication monitoring, SM dependency, and the mitotic spindle checkpoint. So first, we would need to go over good background information regarding the eukaryotic cell division. And first, the cell division occurs in well-defined stages. And these defined stages are known as first G1, then the S phase, G2, and the M phase, those that are underlined there in that first bullet point. In regards to the G1 phase, this is the waiting period that occurs, and no DNA synthesis takes place here, but the RNA and protein synthesis within the cell, this is occurring. But in regards to DNA synthesis, this does not occur in that G1 phase. This is just where the cell is waiting and preparing its way to go throughout the cycle. Now, the next phase, which would be the S phase, known as the synthesis phase, this is where DNA is produced, and the synthesis takes place, where it makes copies for both of the daughter cells. And then the next phase, the G2 phase, this is indicating the gap between divisions here. The G2 phase is where the new proteins are synthesized, and this is where the cell needs to double its size. And the reason that the cell needs to double its size, and we'll get into this more along in the factors, but that cell has to be large enough to begin dividing. If it is too tiny and we don't have a checkpoint there to check it, then it's going to keep dividing and be too tiny for its contents, for the genetic material. So this is crucial that the cell doubles in size before it divides. And then the next phase, which is M phase, this is known as mitosis. And this is where the nuclear division and cytokinesis takes place. Okay? And the mitosis is actually composed of the prophase, the metaphase, the anaphase, and the telophase. And then at the end, it results in cytokinesis, where it has the splitting and the yielding of the two daughter cells. Now next, here, this is the cell cycle and its specific checkpoints here. And the first checkpoint, if you'll look here through the different phases we had, okay, this is that G1 phase. This is that waiting period that the cell has. Okay, right here, if you'll look, this is a cell growth checkpoint. So before the cell ever gets to S phase, it has to go through this growth checkpoint. If it's good to go, then that cell will just keep going into the S phase. But once it gets past this restriction point here, this particular point where it restricts the cell going farther if it isn't prepared, once it gets past that restriction point, it cannot go back. It is fully committed to going throughout the process, and it is committed to go through DNA synthesis, which would occur at the S phase. So after it goes through this cell growth checkpoint, it'll move on to the S phase, and this here is the DNA synthesis checkpoint. This is another checkpoint that is needed for that cell to go through. If it's not ready, then the cell will say, hey, I'm not ready yet. And then it will then prepare itself, okay? Especially if DNA is damaged, which we'll get to. If the DNA is damaged in a cell, it needs to stop, fix that damage before it moves on. And then here is that G2 phase. And then right here at the M phase with mitosis, there's another checkpoint, which is the mitosis checkpoint. Okay? And this is where that cell stops, make sure that it's good to go in regards to the mitosis process. So the prophase, the metaphase, the anaphase, and the telophase. And this is just a few examples of how the cell constantly regulates itself throughout the cycle. And still, in regards to the eukaryotic cell division background, after passing through mitosis and into the G1 phase, a cell has to make a choice. The cell either continues through another division or it's going to cease to divide. And if it does that, it's going to enter that G0 phase that you saw. That was kind of that circular motion in that picture just now. Okay, so a cell has to make a choice, okay, just like people make a choice. And when a cell in the G0 phase, once it decides it wants to begin dividing again, it will re-enter the division cycle through the G1 phase, and then it'll start the process again. Okay, so cells have to make a choice. And if you'll look here at the pictures below, these are just examples of the specific cycle that the cells use to divide. So this first one here, the very first one, that's the interphase, okay? So the interphase includes the G1, the S phase, synthesis phase, where the DNA is replicated and synthesized. This next one is the prophase. Okay, this next one here, this is before metaphase. Okay, this is the pre-metaphase right here. Okay, and then next, this is metaphase. So this is where they're lined up on that metaphase plate. You've heard that. Next, this is the anaphase. Okay, so they're no longer on the plate. They're going to go to both ends of the cell. And then this one here results in the telophase, or the cytokinesis at the end, and this is where it splits into the two daughter cells. Now, in regards to checkpoints in the cell cycle, okay, the progression through the cell cycle division, okay, it is driven by things such as the activation and the inactivation of the cyclin-dependent kinases. And these are known as the CDKs that we'll refer to. And these trigger the transition into subsequent phases of the cycle. And before I go farther, you might ask, well, do all of these cells necessarily go through this? And there's actually a few exceptions. So for example, there are specific differentiated cells. And a few of these are first known as liver cells, is an example of these. The hepatocytes, okay, they are an example, or the adipocytes. They are the fat cells that you find in the connective tissue. And what these cells do is they have a specialized function and forms. And what they're going to do is they're not going to necessarily go through those, that G1, the S phase, G2 phase, and is regulated. What they're going to do is they're just going to constantly stay in that G0 phase is what they're going to do. So there are a few exceptions to these rules. Okay? But in regards to the CDKs, okay, whenever we define them, they are the small serine or therine protein kinases. And that what they do is they require the association with the cyclin subunit for their activation. And this is important with regulating the cell cycle again. And there are many levels of regulation that impinge upon the CDKs to impose tight control over the cell cycle progression. And now we're going to get into the particular things that constant things that regulate the cell cycle. And such regulation involves controlled expression and destruction of cyclins, the activating and inhibitory phosphorylation and dephosphorylation of the CDKs, and expression and destruction of the inhibitory proteins that associate with the CDKs or the CDK cyclin complexes. Now, in regards to different regulators throughout the cell cycle, first, we already talked about these, the CDKs and the cyclins. They play a very important role as regulators in specifically the G1S phase and the G2M transitions. This is where they are important. And we'll also look at a diagram that shows us specific CDKs and cyclins that regulate each part of the cell cycle. Next, we would have the CDK inhibitory proteins. And these are specific protein inhibitors, and they're going to bind and inactivate specific CDKs within the cell cycle if needed. And an example of one of these, it's called P21, P21. It's an example of a CDK inhibitory protein. Okay, next we have the RB-E2F pathway. So this is a tumor suppressor of PRB. It is a well-known transcriptional repressor of genes involved in the S-phase progression, such as specifically in cyclin E. And the proteins of the PRB family, they exert this role through the association with transcription factors in particular. Okay, again, it's another way that the cell regulates throughout the entire cycle. Next, we would have the positive and negative phosphorylation of CDKs. This is the specific targets that are phosphorylated by cyclin CDKs. And these specific targets that are phosphorylated by the cyclin CDKs in particular, they are actually the proteins of the nuclear envelope of the cell, and they are proteins that are required for cytokinesis to happen and DNA repair. Okay, so again, cytokinesis is whenever the cell is going to split into the two daughter cells, and DNA needs to be repaired within the cell. If something is wrong with the DNA of a cell, but it still goes through the cycle and it's not regulated, then you're going to end up with things such as mutations. You're going to end up with too many chromosomes. You're going to end up with not enough chromosomes. You'll lead to diseases, cancers, things of that nature. And finally, the other regulator is the protein degradation. And this is marked by the attachment of ubiquitine to the amino group of a side chain of a lysine residue. Okay, and in particular, ubiquitine. Okay, in case you were wondering, it is a small amino acid. It's composed of 76 amino acids. It is a regulatory protein. And what it does is it's going to direct the movement of important proteins throughout the cell. And in particular, it can be involved in both the synthesis of new proteins that are needed for the cell, or they can be involved in the destruction of proteins that is going to infect the cell, things that are defective for the cell, those defective proteins. All right, and just for another way for you to visualize it, this is an example of the cyclin CDKs throughout the cell cycle, okay? And for example, these ones would be needed, okay, to regulate throughout the G1 phase. Okay? And again, before we get to S phase, before that cell commits to the S phase, it has to go through that restriction point, okay? This restriction point is needed. The cell's got to be large enough, got to be good to go before it begins replicating the DNA here. Okay, these are the CDKs and cyclins needed here. Okay, this is that checkpoint where the cell gives a thumbs up or a thumbs down as to whether the DNA synthesized, it's damaged, or it's good to go, something of that nature. Okay, G2 phase. Again, this is whenever that cell, it has to double its size so it can divide correctly. Okay, and then this M phase, it's got that mitosis checkpoint with it. So again, many things here, such as the cyclins and CDKs and other factors that are going to help that cell just constantly be checking and balancing itself, basically. Okay. So now we're going to get into what are the particular factors that help throughout the cell cycle. And the first one is the cell size control. In order to maintain the cell size, the cells must exactly double their contents before they divide. So you think, why do they absolutely have to exactly double in size before they can even divide? They have to do this to ensure that each daughter cell is supplied with the appropriate amount of DNA material, of genetic material, biosynthetic material. That cell has to be large enough for the appropriate amount of the DNA material for that cell to keep going. That is why that cell has to become doubled in size for it to go throughout this process. And cell size checkpoints that we just saw earlier, they allow the cell to coordinate cell size with the cell cycle progression. The cell size checkpoints are observed, which we already saw this in G1 and G2. There were particular checkpoints within the cell cycle there that either gave, again, thumbs up or thumbs down as to whether that cell was good to go on to the next phase or not. And we see evidence of these checkpoints contained in observations that the size of the new daughter cells after mitosis affect the cell cycle progression. And the evidence that we see is that the large daughter cells, they actually speed up their progress within the cell cycle, especially through the G1 and the G2 phases where you got those cell size checkpoints. So they give a thumbs up automatically and say we're good to go, and they keep going. But the small daughter cells, they don't speed up their progress in the cell cycle. What they do is they delay their exit through those growth phases in G1 and G2. They're not large enough. The cell basically has a red flag going saying, hey, I'm not large enough. Let's stay in this phase. Let's stay here. Let's get larger before we can move on to the next phase. The next factor would be DNA damage responses. And so throughout interphase, the DNA damage elicits a cell cycle arrest. So that cell cycle stops, puts a hold on everything, and says DNA's damaged. We have to do something. And this allows for more time to repair specific pathways to operate before it commits to the other phases of the cell cycle. And DNA damage checkpoints, they can be separated into specific categories. First, they can be separated into those controlled by tumor suppressor and transcription factor P53, or they can be categorized under those that control of the checkpoint kinase that's known as CHK1. And what's important about CHK1 is it can be activated by all known forms of DNA damage, which is crucial. So any type of DNA damage that happens, CHK1 is going to act upon it and it's going to fix it before it moves on to the next phase of the cell cycle. And in higher organisms, in particular, the transcription factor P53 is critical in DNA damage checkpoints. Okay? And what it does, while it's critical in this, it's regulated by a plethora of post-translational modifications. Next, another factor is monitoring DNA replication. So DNA replication is initiated at specific sites in the cell cycle, and they are defined by a number of proteins that ensure that they fire only once per cell cycle. When we say fire, we mean that they just start replicating only once per a cell cycle. And replication origin firing, it is controlled by the phosphorylation of two proteins. These two proteins are known as CDT1 and CDT6, which is catalyzed by both CDKs and DBF4 dependent protein kinase, which that's a mouthful. It's basically known as DDK CDC7. And the effector kinase in particular, this one's known as intra-S phase checkpoint. It is known as CDS1 in fission yeast cells and CHK2 in humans. Okay, and then at that point above, when we're talking about the phosphorylation, this phosphorylation, it not only initiates the replication, but it also leads to the degradation of these proteins. And then what happens there is the origin cannot refire. So it only wants, what we're wanting to do is that cell is only wanting to start replicating that DNA once per cell cycle. And these factors are put into place to make sure that happens. Next is the other factor is SM dependency. And upon stabilization, the replosome, which is the polymerase and associated proteins, they may stay in position until a blockade's removed. So something might be wrong with the cell in regards to DNA replicating and being synthesized at the S phase or at the mitosis phase where that cell is dividing. So this is what is needed to have a checkpoint in to make sure that the cell is good to go, it can keep going. And the cell can employ post-replication repair pathways to bypass any lesions that it has. And it does that through either recruiting mutagenic bypass polymerases, okay, or switching templates by the recombination and then replicating using another nascent strand. Okay? And the checkpoints, they have to be employed here to ensure that mitosis is not attempted until the DNA replication is complete, or the cells are going to run into a risk of reduced ploidy. So an example of this would be Down syndrome, where you don't have enough number of chromosomes. I believe it's specifically in chromosome 21. Don't quote me on that. Don't know at the top of my head. But you might have an example of this to where if the DNA is replicating, okay, and it's not ready for that, it's damaged, it doesn't have enough chromosomes, you have too many chromosomes, the DNA is damaged, it has to make sure that it is only replicated once in order for mitosis to go through and for that cell to divide. Right. And that last factor that we're going to look into, this is the mitotic spindle checkpoint. This is the segregation of sister chromatids at the anaphase stage, and it's under the mechanical control of the mitotic spindle. Okay? And it is important that the spindle attachment, it has to occur in a bi-oriented fashion. And the reason that we're doing this is that the sister chromatids have to be under tension at the metaphase stage before they ever go into anaphase. They have to be in tension under the metaphase stage. And they have to be attached to both poles of the spindle to keep going. They have to be in this bi-oriented fashion under tension. And the cell regulates that and checks for that. And once all the kinetochores are attached and they're aligned at the metaphase plate, then anaphase can proceed, as is promoted, by the activation of a large E3 ubiquitine ligase. And this is a mouthful, but this ligase, it is known as the anaphase promoting complex of cyclosome, or APC-C is what this is known as. And the spindle checkpoint, what its function is it functions to prevent the activation of APC CDC20. Okay? And this is under the conditions where the kinetochores are not occupied by the spindle microtubules or are attached, but they're not necessarily under tension. So again, the cell's checking for that. It's checking to make sure that it is good to go in the metaphase stage once they are lined up on the metaphase plate, the sister chromatids, to where it's good to go on the anaphase stage, where then they go on separate sides of the cell. Okay, so in conclusion, this is a good way to wrap up a presentation to then review with your students what they learned, to make sure they knew what was expected of them, that they went through all the learning goals, which we did. So first, the cell division, and this is accompanying mitosis in eukaryotic cells. It occurs in well-defined stages. Again, these stages are G1, S. S is the synthesis stage where the DNA replication takes place. G2 and M. And M is the mitosis phase. This is where the this is where the cell is dividing. And then the mitosis phase is broken into prophase, metaphase, anaphase, telophase, which then results in cytokinesis, where that cell splits into the two daughter cells. The other part we talked about was the progression through the cell division cycle. It's driven by activation and inactivation of those CDKs, those cyclin-dependent kinases, which trigger the transition to the subsequent phases of the cycle. That's another way that the cell's regulated throughout the cycle. Another thing we talked about was different regulators of the cell cycle, which again included those CDKs and the cyclins, the CDK inhibitory proteins. P21 was an example, the RBE2F pathway, the positive and negative phosphorylations of the CDKs, and then that ubiquitine, the protein degradation. Okay, the ubiquitine, it could either synthesize the good proteins, get rid of those bad ones for the cell. And then the other part we talked about, Another learning goal was that there were important factors of the cell cycle regulation. The first one was cell size control. The cell has to double its contents. The DNA damage response, okay? That cell has to respond to any DNA damage that occurs within the cell. Okay, so no diseases, no cancers come about with it. DNA replication monitoring, that we only want replication to happen once. SM dependency. Okay, again, before that cell goes into mitosis, we only want the DNA to be replicated and synthesized in the S phase just once and has to be good to go, or the mitotic spindle checkpoint. Okay. So I hope that you enjoyed this presentation. Please leave me comments as to how I could have improved this. Thank you very much. I hope everyone has a wonderful day. These are my sources, my reference list, if you would like to check any of them out. Thank you.